Since the DNA molecule is trapped within the cell nucleus because it's very big, it's the entire genome, it is double-stranded helix, so it's very big, it cannot leave the nucleus, it cannot pass through the nuclear pores in the nuclear envelope. So there has to be another molecule that carries the message or the instructions from the DNA molecule to the ribosomes where protein synthesis takes place on the rough ER. How would this genetic information get carried from the nucleus to the cytoplasm? Through the RNA molecule. So that makes us come to a conclusion that RNA molecule is shorter. It will only carry information for one protein that is being instructed. So if my cell needs enzyme A, then only that part of the DNA that is the gene for enzyme A is translated to an RNA message. So that's why the RNA molecule is shorter, because again it represents only a portion of the DNA molecule. Not only that, it is also single-stranded, so it's shorter and smaller in diameter. It's a single-stranded molecule, single polynucleotide, and hence it can cross through the nuclear pores to the cytoplasm. Before we see how this RNA molecule copy the information from the DNA and then carry it to the cytoplasm, let's see the three types of RNA molecules. So the RNA molecule is further subdivided into three types. mRNA, messenger RNA, carry the message. rRNA, ribosomal RNA, so this is part of the ribosome, whether they are free or on the rough ER. tRNA, transfer RNA, and this RNA transfers the amino acid to the newly formed protein. So messenger, carry the message from the DNA. Ribosomal, responsible for reading the message and sensitizing the protein since it is part of the ribosome. Transfer is responsible for bringing the amino acids to the ribosomes where protein synthesis takes place. And we already talked about the differences between DNA and RNA. Let's see how the three types of RNA synthesize protein. As you see here, this yellow part is a cytoplasm. This blue part is the nucleus. So we have in the nucleus a DNA double helix with all the genetic information. When it's time to synthesize a protein, the DNA strands pulls apart only at the site where the gene for that protein is found. So this is the part that carries the gene for the specific protein that we are synthesizing. So what happened is a new mRNA, messenger RNA, starts to form, completing the opposite strand. Why completing the opposite strand, not this one? Because once it completes this opposite strand, it will be identical to this one, and so it will carry the message. So if this is the gene, the mRNA will form on the opposite one, so that it will be complementary bases, carry the same bases similar to the gene. Okay, so again, DNA only splits at the part of the gene. That's the first step. Information is copied, and copying the information is called transcription. Copy of the information from the DNA molecule to the mRNA molecule is called transcription. So, as a definition, transcription is the formation of mRNA molecule from the DNA molecule. That's transcription. And it takes care inside or is carried out inside the nucleus. So let's take a closer look here. As you see, A complements the T. U complemented the A. Why U? Because I'm forming RNA. I'm not forming TNA. So U here will be placed in front of the A. Yes, the message is being read from the DNA, but it is translated or transcribed into mRNA. So if I give you in the test the DNA molecule or the genetic code or nucleotide bases on the DNA molecule and ask you to tell me the complementary DNA, you will use a T. If I give you the DNA nucleotides and ask you to give me the messenger RNA, you will use a U. So if I am giving you C, look down here, C, G, T, C, C, G, A, G, and ask you to form an mRNA, so you'll put G, C, A, G, G, C, and in front of the A, that's the trick here, you will put U, because this is an RNA, and then again C. But if I'm giving you the DNA and ask you to give me the complementary here, so you will go the same way as we're going, and once you arrive at the A, you will put a T here instead of that A. Now let's get back to protein synthesis. So we formed, or the cell or the nucleus formed the messenger RNA from the DNA, and that's called transcription. Then this short messenger RNA, short and single-stranded, can pass through the nuclear pores and go and attach itself to the ribosomes. This brown is the ribosome. Ribosome on the rough ER or free ribosomes, it will just attach itself to the ribosome. In the ribosome, this is the rRNA. The message from the mRNA is going to be read. So the ribosome will start reading the message from the mRNA. And as it reads the message, it will translate it to amino acid. How is that? The ribosome will start to read each 3 and bring the opposite 3 here. So let's give it names. Each 3 nucleotides on the messenger RNA, we will call them codon. Codon. Why did we say 3? Because if you recall, each 3 nucleotide represent 1 protein. So again, each 3 nucleotides on the messenger RNA is called a codon. It codes for 1 amino acid, as you see here. But this codon has to be read by the ribosome and translated by the transfer RNA. Those are the transfer RNA. They start to bring the amino acids carried on them. If you, again, I told you the tRNA or the transfer RNA bring amino acids according to the message. So they have to match. Okay? They bring the amino acid, add it to the chain, and then they leave empty. And then the next transfer RNA carrying the next amino acid. This is the anticodon. Anticodon. So the anticodon is part of the transfer RNA. It matches with the codon on the mRNA. If it matches, it lets go of the amino acid. So let's go over it again. DNA strand pulls apart or split where the gene for that specific protein is present. DNA information will be copied or transcribed into mRNA. So transcription means the formation of mRNA from DNA molecule inside the nucleus. The mRNA leaves the nucleus and attaches itself to the ribosome. Once it attaches to the ribosome, translation begins, meaning that the ribosome will start reading that message. Translation is reading the message on the mRNA and start to forming the amino acid chain. So translation again begins as the transferred RNA with their anticodon, these three, recognize the mRNA codons and thus bring the correct amino acid into position in the growing polypeptide chain. Once translation begins, it keeps going until the whole message is being read. So as the ribosome moves along the mRNA, more amino acids are being added. So if I ask you here, what is the ribosome doing? It is reading the message on the mRNA. And as it's reading it, it is reading it in 3 nucleotides at a time. So it reads this first nucleotide, send the message to the matching tRNA and bring the matching amino acid. Then at the end of the mRNA ribosome, the ribosome will release the new protein. So once the message is finished, the ribosome will release that newly formed protein, and those free transfer RNA or tRNA can go and pick another amino acid to be reused. So the tRNA are recyclable. What do you need